In molecular biology, the term double helix refers to the structure formed by double-stranded molecules of nucleic acids such as DNA. The double helical structure of a nucleic acid complex arises as a consequence of its secondary structure, and is a fundamental component in determining its tertiary structure. The term entered popular culture with the publication in 1968 of the Double Helix, a personal account of the discovery of the structure of DNA by James Watson. The DNA double helix polymer of nucleic acid, held together by nucleotides which base pair together. In DNA, the most common double helical structure found in nature, the double helix is right-handed with about 10 to 10.5 base pairs per turn. This translates into about 20-21 nucleotides per turn. The double helix structure of DNA contains a major groove and minor groove. In DNA, the major groove is wider than the minor groove. Given the difference in widths of the major groove and minor groove, many proteins which bind to DNA pass through the wider major groove. History. The double helix model of DNA structure was first published in the journal Nature by James Watson and Francis Crick in 1953, based upon the crucial X-ray diffraction image of DNA labeled as Photo 51 from Rosalind Franklin in 1952, followed by her more clarified DNA image with Raymond Gosling, Maurice Wilkins, Alexander Stokes, and Herbert Wilson, and base pairing chemical and biochemical information by Erwin Chargaff. The prior model was triple-stranded DNA. The realization that the structure of DNA is that of a double helix elucidated the mechanism of base pairing by which genetic information is stored and copied in living organisms, and is widely considered one of the most important scientific discoveries of the 20th century. Crick, Wilkins, and Watson each received 1/3 of the 1962 Nobel Prize in Physiology or Medicine for their contributions to the discovery. Nucleic acid hybridization. Hybridization is the process of complementary base pairs binding to form a double helix. Melting is the process by which the interactions between the strands of the double helix have broken, separating the two nucleic acid strands. These bonds are weak, easily separated by gentle heating, enzymes, or physical force. Melting occurs preferentially at certain points in the nucleic acid. T and A-rich regions are more easily melted than C and G-rich regions. Some base steps are also susceptible to DNA melting, such as TA and TG. These mechanical features are reflected by the use of sequences such as TATA at the start of many genes to assist RNA polymerase in melting the DNA. For transcription, strand separation by gentle heating, as used in polymerase chain reaction, is simple, providing the molecules have fewer than about 10,000 base pairs. The intertwining of the DNA strands makes long segments difficult to separate. The cell avoids this problem by allowing its DNA melting enzymes to work concurrently with topoisomerases, which can chemically cleave the phosphate backbone of one of the strands so that it can swivel around the other. Helicases unwind the strands to facilitate the advance of sequence reading enzymes such as DNA polymerase. Base pair geometry. The geometry of a base, or base pair step, can be characterized by 6 coordinates: shift, slide, rise, tilt, roll, and twist. These values precisely define the location and orientation in space of every base pair in a nucleic acid molecule relative to its predecessor along the axis of the helix. Together, they characterize the helical structure of the molecule. In regions of DNA or RNA where the normal structure is disrupted, the change in these values can be used to describe such disruption. For each base pair considered relative to its predecessor, there are the following base pair geometries to consider: shear, stretch, stagger, buckle, propeller, rotation of one base with respect to the other in the same base pair, opening, shift, displacement along an axis in the base pair plane perpendicular to the first, directed from the minor to the major groove, slide, displacement along an axis in the plane of the base pair directed from one strand to the other, rise, displacement along the helix axis, tilt, rotation around the shift axis, roll, rotation around the slide axis, twist, rotation around the rise axis, x displacement, y displacement, inclination, tip, pitch, the number of base pairs per complete turn of the helix. Rise and twist determine the handedness and pitch of the helix. The other coordinates, by contrast, can be 0. Slide and shift are typically small in DNA but are substantial in E and Z-DNA. Roll and tilt make successive base pairs less parallel and are typically small. Note that tilt has often been used differently in the scientific literature, referring to the deviation of the first interstrand base pair axis from perpendicularity to the helix axis. This corresponds to slide between a succession of base pairs, and in helix-based coordinates is properly termed inclination. Helix geometries. At least 3 DNA conformations are believed to be found in nature: A DNA, B DNA, and Z DNA. The B form described by James Watson and Francis Crick is believed to predominate in cells. It is 23.7 A wide and extends 34 A per 10 BP of sequence. The double helix makes one complete turn about its axis every 10.4 to 10.5 base pairs in solution. This frequency of twist depends largely on stacking forces that each base exerts on its neighbors in the chain. The absolute configuration of the bases determines the direction of the helical curve for a given conformation. The DNA and Z-DNA differ significantly in the geometry and dimensions to be DNA, although still form helical structures. It was long thought that the A form only occurs in dehydrated samples of DNA in the laboratory, such as those used in crystallographic experiments, and in hybrid pairings of DNA and RNA strands, but DNA dehydration does occur in vivo, and DNA is now known to have biological functions. Segments of DNA that cells have been methylated for regulatory purposes may adopt the Z geometry, in which the strands turn about the helical axis the opposite way to A DNA and B DNA. There is also evidence of protein DNA complexes forming Z-DNA structures. Other conformations are possible: A DNA, B DNA, C DNA, E DNA, L DNA, P DNA, S DNA, Z DNA, etc. have been described so far. In fact, only the letters F, Q, U, V, and Y are now available to describe any new DNA structure that may appear in the future. However, most of these forms have been created synthetically and have not been observed in naturally occurring biological systems. There are also triple-stranded DNA forms and quadruplex forms such as the G quadruplex. Grooves, twin helical strands form the DNA backbone. Another double helix may be found by tracing the spaces or grooves between the strands. These voids are adjacent to the base pairs and may provide a binding site. As the strands are not directly opposite each other, the grooves are unequally sized. One groove, the major groove, is 22A wide and the other, the minor groove, is 12A wide. The narrowness of the minor groove means that the edges of the bases are more accessible in the major groove. As a result, proteins like transcription factors that can bind to specific sequences in double-stranded DNA usually make contacts to the sides of the bases, exposed in the major groove. This situation varies in unusual conformations of DNA within the cell, but the major and minor grooves are always named to reflect the differences in size that would be seen if the DNA is twisted back into the ordinary B form. Non-double helical forms. Alternative non-helical models were briefly considered in the late 1970s as a potential solution to problems in DNA replication in plasmids and chromatin. However, the models were set aside in favor of the double helical model due to subsequent experimental advances such as X-ray crystallography of DNA, duplexes, and later the nucleosome core particle, and the discovery of topoisomerases. Also, the non-double helical models are not currently accepted by the mainstream scientific community. Single-stranded nucleic acids do not adopt a helical formation, and are described by models such as the random coil or worm-like chain bending. DNA is a relatively rigid polymer, typically modeled as a worm-like chain. It has three significant degrees of freedom: bending, twisting, and compression, each of which cause certain limits on what is possible with DNA within a cell. Twisting torsional stiffness is important for the circularization of DNA and the orientation of DNA-bound proteins relative to each other, and bending axial stiffness is important for DNA wrapping and circularization and protein interactions. Compression extension is relatively unimportant in the absence of high tension. Persistence length. Axial stiffness. DNA in solution does not take a rigid structure but is continually changing conformation due to thermal vibration and collisions with water molecules, which makes classical measures of rigidity impossible. Hence, the bending stiffness of DNA is measured by the persistence length, defined as the length of DNA over which the time-averaged orientation of the polymer becomes uncorrelated by a factor of V. This value may be directly measured using an atomic force microscope to directly image DNA molecules of various lengths. In an aqueous solution, the average persistence length is 46 to 50 nanometers or 140 to 150 base pairs, although can vary significantly. This makes DNA a moderately stiff molecule. The persistence length of a section of DNA is somewhat dependent on its sequence, and this can cause significant variation. The variation is largely due to base stacking energies and the residues which extend into the minor and major grooves. Models for DNA bending. The entropic flexibility of DNA is remarkably consistent with standard polymer physics models, such as the Crick-powered worm-like chain model. Consistent with the worm-like chain model is the observation that bending DNA is also described by Hooke's law at very small forces. However, for DNA segments less than the persistence length, the bending force is approximately constant and behavior deviates from the worm-like chain predictions. This effect results in unusual ease in circularizing small DNA molecules and a higher probability of finding highly bent sections of DNA. Bending preference. DNA molecules often have a preferred direction to bend, i.e. anisotropic bending. This is, again, due to the properties of the bases which make up the DNA sequence. A random sequence will have no preferred bend direction, i.e. isotropic bending. Preferred DNA bend direction is determined by the stability of stacking each base on top of the next. If unstable base stacking steps are always found on one side of the DNA helix, then the DNA will preferentially bend away from the direction. As bend angle increases, then steric hindrances and ability to roll the residues relative to each other also play a role, especially in the minor groove. A and T residues will preferentially be found in the minor grooves on the inside of bends. This effect is particularly seen in DNA-protein binding where tight DNA bending is induced, such as in nucleosome particles. See base step distortions above.
DNA molecules with exceptional bending preference can become intrinsically bent. This was first observed in trypanosomatid kinetoplast DNA. Typical sequences which shows this contain stretches of 4 to 6 DNA residues separated by G and C-rich sections which keep the A and T residues in phase with the minor groove on one side of the molecule. For example, GATTCCAAAAATGCCAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAAA
of base pairs relative to each other, allowing unusual bifurcated hydrogen bonds between base steps. At high temperatures this structure, and so the intrinsic bend, is lost. All DNA which bends anisotropically has, on average, a longer persistence length and greater axial stiffness. This increased rigidity is required to prevent random bending, which would make the molecule act isotropically. Circularization. DNA circularization depends on both the axial stiffness and torsional stiffness of the molecule. For a DNA molecule to successfully circularize, it must be long enough to easily bend into the full circle and must have the correct number of bases, so the ends are in the correct rotation to allow bonding to occur. The optimum length for circularization of DNA is around 400 base pairs, with an integral number of turns of the DNA helix, i.e., multiples of 10.4 base pairs. Having a non-integral number of turns presents a significant energy barrier for circularization. For example, a 10.4 x 30 equals 312 base pair molecule will circularize hundreds of times faster than 10.4 by 30.5 approximately equals 317 base pair molecule. Stretching. Longer stretches of DNA are entropically elastic under tension. When DNA is in solution, it undergoes continuous structural variations due to the energy available in the thermal bath of the solvent. This is due to the thermal vibration of the molecule combined with continual collisions with water molecules. For entropic reasons, more compact relaxed states are thermally accessible than stretched out states, and so DNA molecules are almost universally found in a tangled relaxed layouts. For this reason, one molecule of DNA will stretch under a force, straightening it out. Using optical tweezers, the entropic stretching behavior of DNA has been studied and analyzed from a polymer physics perspective, and it has been found that DNA behaves largely like the Crick-pored worm-like chain model under physiologically accessible energy scales. Under sufficient tension and positive torque, DNA is thought to undergo a phase transition with the bases splaying outwards and the phosphates moving to the middle. This proposed structure for overstretched DNA has been called P-form DNA, in honor of Linus Pauling, who originally presented it as a possible structure of DNA. The mechanical properties of DNA under compression have not been characterized due to experimental difficulties in preventing the polymer from burning under the compressive force. Supercoiling and topology. The B form of the DNA helix twists 360 degrees per 10.4 to 10.5 bp in the absence of torsional strain, but many molecular biological processes can induce torsional strain. A DNA segment with excess or insufficient helical twisting is referred to, respectively, as positively and negatively supercoiled. DNA in vivo is typically negatively supercoiled, which facilitates the unwinding of the double helix required for RNA transcription. Within the cell, most DNA is topologically restricted. DNA is typically found in closed loops which are topologically closed, or as very long molecules whose diffusion coefficients produce effectively topologically closed domains. Linear sections of DNA are also commonly bound to proteins or physical structures to form closed topological loops. Francis Crick was one of the first to propose the importance of linking numbers when considering DNA supercoils. In a paper published in 1976, Crick outlined the problem as follows. In considering supercoils formed by closed double-stranded molecules of DNA, certain mathematical concepts, such as the linking number and the twist, are needed. The meaning of these for a closed ribbon is explained, and also that of the writhing number of a closed curve. Some simple examples are given, some of which may be relevant to the structure of chromatin. Analysis of DNA topology uses three values. L equals linking number, the number of times one DNA strand wraps around the other. It is an integer for a closed loop and constant for a closed topological domain. T equals twist, total number of turns in the double-stranded DNA helix. This will normally tend to approach the number of turns that a topologically open double-stranded DNA helix makes free in solution. Number of bases, 10.5, assuming there are no intercalating agents or other elements modifying the stiffness of the DNA. W equals writhe, number of turns of the double-stranded DNA helix around the superhelical axis. L equals T plus W, and delta L equals delta T plus delta W. Any change of T in a closed topological domain must be balanced by a change in W, and vice versa. This results in higher order structure of DNA. A circular DNA molecule with a writhe of 0 will be circular. If the twist of this molecule is subsequently increased or decreased by supercoiling, then the writhe will be appropriately altered, making the molecule undergo plectonemic or toroidal superhelical coiling. When the ends of a piece of double-stranded helical DNA are joined so that it forms a circle, the strands are topologically knotted. This means the single strands cannot be separated. Any process that does not involve breaking a strand, the task of unknotting topologically linked strands of DNA, falls to enzymes termed topoisomerases. These enzymes are dedicated to unknotting circular DNA by cleaving one or both strands so that another double or single-stranded segment can pass through. This unknotting is required for the replication of circular DNA and various types of recombination in linear DNA which have similar topological constraints. The linking number paradox. For many years, the origin of residual supercoiling in eukaryotic genomes remained unclear. This topological puzzle was referred to by some as the linking number paradox. However, when experimentally determined structures of the nucleosome displayed an over-twisted left-handed wrap of DNA around the histone octamer, this paradox was considered to be solved by the scientific community.